Hello, and thank you for coming to check out my talk. Today I'm going to be telling you about some research from my dissertation in which I look at the telomere dynamics of male wire-tailed manakins. So telomeres are these repetitive sequences of DNA found at the end of eukaryotic chromosomes. So it's this sequence repeated over and over and over again. And I like to think about telomeres as the aglets found on the ends of shoelaces. And the reason being is that telomeres are responsible for protecting DNA and also delineating DNA, or the ends of DNA, during cellular replication. And telomeres are particularly interesting because they change in length over the course of an individual's lifetime. Now, they can increase in length via repair mechanisms such as telomerase, but they also decrease in length due to things like the DNA replication process and oxidative stress. And once telomeres reach or shorten to a certain length, cells enter a senescent state and actually stop replicating. So when whole organs or whole tissues exhibit short telomeres, that's when problems at the level of the organism start to arise. And biologists have identified a number of factors that contribute to rates of telomere shortening, and these include aging. So in general, as individuals age, their telomeres get shorter. An individual's nutritional status can influence rates of telomere shortening. So high antioxidant diets are associated with slower rates of telomere shortening. And then an individual's disease status can also influence the rate that their telomeres shorten. And diseases like avian malaria have actually been found to accelerate rates of telomere shortening. Now, telomere lengths also depend upon an individual's developmental environment. And so the developmental environment can kind of set the stage for an individual's lifetime in terms of their telomere length. And also related to this picture, an individual's reproductive investment can influence the rate that their telomeres shorten. So higher reproductive investments are associated with greater rates of telomere loss. And lastly, aspects related to an individual's environment, such as the land cover or annual variation in climatic factors, can also influence rates of telomere loss. Now, telomeres vary with mortality risk. Specifically, an individual's telomere length, or shorter telomeres, I should say, are associated with higher mortality risks. And the studies I'm including here are not focused on a single species, but rather either represent a meta-analysis showing this pattern, or data from multiple species have identified this pattern. And across species, we also see another pattern in that an individual's or a species' lifespan is related to the rate that telomeres shorten. And what we find in general is that longer lifespans are associated with shorter or slower rates of telomere attrition. And there are a number of other factors that vary with lifespan, and these include things like reproductive output for each reproductive event, or an individual's reproductive investment, that is, metabolic rates and annual survival, of course, and the framework that is kind of developed in order to understand these patterns is the pace of life framework. And so this pattern here between telomere shortening and lifespan also integrates telomeres into this suite of traits that vary along this slow to fast-paced continuum. And within and between species, what we predict is that individuals or species with a slower pace of life exhibit slower rates of telomere shortening. Now, because telomeres integrate information about an individual's environment and physiology, and also relate to their mortality risk, telomeres are thought to represent an individual's biological age. So by measuring telomere lengths or changes in telomere lengths, we can understand the causes and consequences of biological aging. Now today I want to talk to you about the telomere dynamics of a species of manakin. And so manakins are a super cool family of birds. There's an entire symposium at SICB dedicated to them. I encourage you to check out all the other talks. Maybe that's how you found your way here. So this slide here is showing you various different species of manakins. But there's one caveat in that these are only the males. And so the males in this species are bright and flashy, and they have all these cool courtship displays. And, it's a lek mating system, so they don't contribute anything to parental care. But the species I'm going to talk to you about today is the wire-tailed manakin. And the wire-tailed manakin has been the subject of a long-term study initiated by Brent Ryder that has occurred in northeastern Ecuador. And one of the things we know from this long-term data set is that wire-tailed manakins have an exceptional lifespan for their size. And so they're about the size of a chicken egg, but their average lifespan is almost 9 years. And their maximum lifespan, we don't even know what their maximum lifespan is because the oldest individuals in the population are still alive. Okay, so we're at 16, so it's somewhere over 16. And then another study that I want to highlight, they looked at, they compared an assortment of species found in the Peruvian Amazon. They measured metabolic rates, and then they did band recapture studies, mark recapture studies to look at survival. And what they found was that out of all the species they sampled, a sister species to the wire-tailed manakin, that's the band-tailed manakin, exhibited the lowest metabolic rate and the highest annual survival. And so what I'm trying to show you here is that manakins exhibit slow paces of life, and especially for their size, they exhibit exceptional lifespans. And so to look at the telomere dynamics, we sampled males. We collected blood samples over the course of 4 years. We measured telomeres in the blood using qPCR, telomere lengths. And in the analyses I'm going to talk to you about today, we only included known age males, so males of known chronological age. And here's a histogram of our data set. You can see that our samples spanned quite the age range. The color indicates an individual's social status. I'm not going to talk about that too much today. Only the dark bars represent individuals that are reproducing in the population, territory holders. But in general, we had 187 samples from 53 males. So there's quite a bit of resampling in this study that I'm working with. And if we look at the relationship between age and telomere length, what we find is that there is basically no pattern. And to analyze these data, we broke it down into between and within individual effects. So this average age term is a between individual effect. This is a within individual changes. And like I said, there's no consistent pattern in age-related changes in telomere lengths within or between individuals. Now, another way to look at the stability of telomere lengths is using this repeatability estimate. And so repeatability is an intraclass correlation coefficient, technically speaking. And what it represents is how correlated are an individual's measurements. In other words, it's the amount of variation that's due to between, as opposed to within individual differences. And so as you can see in this graph, out of the species for which these data are available, wire-tailed manakins exhibit exceptionally stable telomeres. And this is likely related to their extreme lifespan for their size, and also related to the fact that manakins are highly frugivorous, and fruit-based diets are associated with high antioxidants. So it's likely that their diet is also involved in driving the stability of the telomere dynamics in this species. So a quick conclusion here, male wire-tailed manakins do not exhibit age-related changes in telomere length, and we looked at this both within and between individuals, and there were no consistent patterns. And then wire-tailed manakins also exhibit exceptionally stable telomeres. Now there's another question that I would like to introduce, which is how does an individual's telomeres relate to male behavior? And so behavior is another trait that's involved in the pace of life framework. And in general, what we predict is that individuals with a slower pace of life exhibit lower reproductive investments. And to bring telomeres into this, we would expect that individuals with longer telomeres to exhibit a slower pace of life, and individuals with shorter telomeres to exhibit a faster pace of life or higher reproductive investments. And so we would also make some predictions about an individual's investment in self-maintenance, which is reflected by the changes in telomere dynamics. And so higher reproductive investments would be predicted to be associated with faster rates of telomere shortening. And that's exactly what we found. So we addressed how do telomere lengths relate to male behavior, and then does male behavior relate to variation in telomere dynamics? And behavior in this case is reflective of an individual's reproductive investment, because the males basically don't do anything but court females. They don't help with parental care. They don't help build the nest. They don't take care of the female when she's incubating. They don't do anything but court females and hopefully copulate if they're a winner. So what we found in this study, though, which is included in the files on the Pathable website, what we found is that more social males exhibit shorter telomeres and faster rates of telomere attrition. So like I said, this study not only describes the telomere dynamics, or the unique telomere dynamics in this species, but it also shows how male behavior, which is related to their pace of life, relates to both their telomere length and the changes in telomere length. So check it out more if you're interested. You can download the paper directly from the conference website in this talks page. With that, I would like to thank all the staff and management at Tipitini for helping us with this work, helping us with all the logistics, the various people who contributed to us collecting these data, and the development of the telomere assay. I would like to acknowledge some folks at Virginia Tech, and then of course the funding, the American Ornithological Society, National Science Foundation, and the Interfaces of Global Change program at Virginia Tech. Each of these organizations helped fund this research. And with that, I will leave you with my contact information as well as my website. Check it out to learn more about my other research. You can also check me out on Twitter. Please reach out if you want to talk about any of this more. Love to hear what your thoughts. And thank you for listening, and I hope you have a good conference.